Welcome back to the third instalment of the Bridging series, where together we consolidate the key GCSE knowledge needed to give you a head start in A-level biology. And we'll send you into the course with secure foundations ready to build on. In this video, we're going to focus on the core GCSE knowledge you'll need for Year 12 on the topics of digestion, gas exchange, the cardiovascular system, and plant transport. We'll also throw in a few pieces of A-level content to whet your appetite for diving deeper as your A-level journey begins. Before we start, do have a look at the free downloads in the description, including a summary of the common misconceptions and errors students make on these topics. The core aim of this channel is to help you understand where you're losing marks, and more importantly, how to avoid making those mistakes in the future. If I can help you avoid the misconceptions that catch out thousands of students every year, you'll walk into your exams with greater confidence and give yourself the best chance of achieving the grades you're aiming for. So let's get started with digestion. You'll remember from GCSE that digestion is the process of breaking down large insoluble molecules into smaller soluble molecules so that they are small enough to be absorbed through the wall of the small intestine and into the bloodstream. This breakdown begins with mechanical digestion, where food is physically broken into smaller pieces in the mouth. Your teeth increase the surface area of the food, making it easier for enzymes to act. In the stomach, powerful muscular contractions churn the food, mixing it thoroughly with digestive juices. Alongside this is chemical digestion, carried out by enzymes. And you'll already know that enzymes are biological catalysts made of protein. They speed up reactions without being used up themselves, and they have a very specific shaped active site that is complementary to the substrate they act on. At A level, you'll continue to study exactly how many more enzymes work, but for now, it's important to remember which enzymes digest which food groups. Carbohydrates are broken down by carbohydrases, such as amylase, into simple sugars. Protein are broken down by proteases into amino acids, and you might have heard of pepsin, while lipids are broken down by lipases into fatty acids and glycerol. One of the biggest misconceptions students bring from GCSE is thinking that enzymes only break substances down, but you will learn at A level that enzymes have an important role in building larger molecules after those small molecules have been absorbed. And another point worth reinforcing is that digestion and absorption are not the same process. Digestion is the breakdown of molecules, whereas absorption is the movement of those small soluble products across the epithelial cells of the small intestine into the blood or lymph fluid. Keeping those two ideas separate will help you avoid a surprisingly common exam error. As we move into A-level biology, you'll also continue to understand why the small intestine is so effective at absorption. Remember the three main factors affecting rate of diffusion: large surface area due to villi and microvilli, a thin layer of single cells reducing the diffusion distance, and a good blood supply to maintain the concentration gradient. You'll go on to learn how epithelial cells are adapted with important proteins and organelles for transport, and how, while some nutrients enter by diffusion, others require other processes. Now, with that foundation in place, let's move on to gas exchange and remind ourselves how the lungs are adapted to supply oxygen for aerobic respiration. So, the gas exchange system not only supplies oxygen for aerobic respiration, it has to remove carbon dioxide, which is a produced waste product of respiration. The lungs contain millions of tiny air sacs called alveoli, and it's here that gas exchange takes place by diffusion. Oxygen diffuses from the air inside the alveoli into the blood, while carbon dioxide diffuses in the opposite direction from the blood into the alveoli, ready to be exhaled. At GCSE, you will have learnt the key adaptations of the alveoli. They provide a large surface area, have thin walls, are moist, and are surrounded by an extensive capillary network. These are still absolutely essential at A level. For example, the millions of alveoli collectively provide a huge surface area, allowing more oxygen molecules to diffuse into the blood every second. The walls of both the alveoli and the capillaries are only 1 cell thick, creating a very short diffusion pathway. Moist surfaces allow oxygen to dissolve before diffusing across the cell membranes. And the rich blood supply continually removes oxygen from around the alveoli while bringing more carbon dioxide. And this helps to maintain a steep concentration gradient, which is one of the most important factors affecting the rate of diffusion. In fact, concentration gradients are something you will encounter repeatedly throughout A-level biology. Whether you're studying gas exchange, transport across cell membranes, or photosynthesis, you'll find yourself returning to the same underlying principles. Another important adaptation is ventilation. Every breath you take replaces the air in the alveoli with fresh air containing a high concentration of oxygen and a lower concentration of carbon dioxide. At the same time, blood flowing through the capillaries continually delivers carbon dioxide and carries oxygen away. Together, ventilation and blood flow work to maintain those steep concentration gradients that make diffusion efficient. You should be able to recall the physical processes that act to ventilate the lungs, breathing. You will need to know this at A level, so make sure you can describe the following steps. The diaphragm, which is made of muscle, contracts and moves down. The intercostal muscles between the ribs contract and move the rib cage up and out, and this increases the volume of the chest cavity. This lowers the pressure in the chest cavity below air pressure, so air moves down a pressure gradient into the lungs. The reverse process happens when breathing out, starting with the relaxation of the diaphragm to move it up, and this starts to decrease the volume of the chest cavity. Now, one misconception that frequently catches students out is confusing ventilation, breathing, gas exchange, and respiration. These are completely different processes. Ventilation is simply the movement of air into and out of the lungs. Breathing is the physical processes that lead to ventilation. Gas exchange is the diffusion of oxygen and carbon dioxide between the alveoli and the blood. And respiration is the series of chemical reactions that release energy inside cells. If you can keep those ideas separate from the very beginning of Year 12, you'll avoid some of the most common mistakes we see in exam answers. Finally, remember that oxygen simply doesn't diffuse into the blood and stop there. It binds to hemoglobin inside red blood cells, allowing large amounts of oxygen to be transported around the body to respiring tissues. The red blood cells, remember, have a large surface area to volume ratio through taking a shape of a biconcave disc, and they have no nucleus to make extra space for oxygen carriage. Understanding exactly how hemoglobin loads and unloads oxygen is another fascinating topic you'll meet later in Year 12. So before moving on, make sure you're confident with these ideas. Next, let's turn our attention to the cardiovascular system and remind ourselves how oxygen, carbon dioxide, and other substances are transported around the body. At GCSE, you learn that the cardiovascular system delivers oxygen and nutrients to respiring cells, removes waste products such as carbon dioxide, and carries many other substances, including hormones and immune cells, to where they are needed. At the centre of this transport system is the heart, which acts as a muscular pump. You'll already know that humans have a double circulatory system, meaning that blood passes through the heart twice during one complete circulation of the body. The pulmonary circulation carries deoxygenated blood from the heart to the lungs, where carbon dioxide is removed and oxygen is absorbed. The oxygenated blood then returns to the heart before being pumped around the body by the systemic circulation. At GCSE, you will have spent time learning the names of the chambers, valves, and major blood vessels, and it's worth making sure you are confident with these before starting Year 12. A key rule is veins take blood into the heart and arteries away from the heart. The vena cava returns deoxygenated blood from the body into the right atrium. The blood then passes into the right ventricle before leaving through the pulmonary artery to travel to the lungs. The prefix pulmonary refers to the lungs. The artery tells us it is taking blood away from the heart. Oxygenated blood will return via the pulmonary vein, pulmonary, lungs, vein, into the heart. The pulmonary vein goes into the left atrium, passes into the left ventricle, and then the blood is finally pumped through the aorta to the rest of the body. You should remember that the left ventricle is thicker than the right ventricle for this reason. It has a thick muscle layer to provide enough pressure for the blood to travel all of the way around the body. And it's the high pressure in arteries that requires thick muscular walls containing elastic tissue to withstand and maintain the high pressures generated by ventricular contraction. Veins have thinner walls and wider lumens because blood is returning to the heart at much lower pressure. And for this reason, they also contain valves to prevent any backflow of blood. Capillaries, however, are made from a single layer of endothelial cells, providing a very short diffusion pathway for the exchange of substances between the blood and surrounding tissues. You will also go on to describe and explain the structure of another type of blood vessel, the arteriole, which are smaller extensions of arteries that control the blood flow into capillaries. Now, notice how we're emphasizing explanations that link structure to function. This becomes even more important at A level. Rather than saying the artery has a thick wall, you'll be expected to explain that the thick muscular layer allows it to contract, and the elastic wall allows it to recoil after stretching to maintain blood pressure. And together, this thick wall enables it to withstand high blood pressure as blood exits the heart. And finally, remember that the heart is much more than a pump. During year 12, you'll study the cardiac cycle, learning exactly how pressure changes move blood through the chambers, how valves open and close, how the heartbeat is coordinated by electrical activity within the heart itself. And you'll also begin calculating cardiac output, interpreting pressure graphs and electrocardiograms, which is a significant step up from GCSE. So, before moving on, make sure you're confident with the pathway of blood through the heart, the difference between pulmonary and systemic circulation, the roles and structures of arteries, veins, and capillaries, and perhaps most importantly, that arteries carry blood away from the heart and veins carry blood towards the heart, regardless of whether the blood is oxygenated or deoxygenated. So, finally, let's turn our attention to plant transport. Although plants don't have a heart or circulatory system like animals, they still need an efficient transport system. Every cell requires water, mineral ions, and sugars, but because plants are fixed in one place, they've evolved a completely different solution to moving substances around the organism. At GCSE, you've learned that plants have two specialised transport tissues, xylem and phloem. The xylem is a dead tissue which transports water and dissolved mineral ions from the roots to the leaves. This movement is one way because water is continually absorbed from the soil and transported upwards to replace water lost from transpiration. The phloem, on the other hand, transports sugars such as sucrose along with other organic substances like amino acids in a process called translocation. The phloem is a living tissue and is made up of sieve tube elements and companion cells, which have a nucleus and mitochondria to provide the energy needed to load sugars from photosynthesis into the phloem. Unlike xylem, transport in the phloem can occur in both directions, depending on where in the plant the sugars are being produced and where they are needed. One of the most common misconceptions students bring into A level is thinking that phloem only transports sugars from the leaves down to the roots. In reality, sugars move from sources to sinks. A source is any part of the plant that produces or releases sugars, such as photosynthesizing leaves, whilst a sink is any region that uses or stores those sugars, such as growing shoots, roots, flowers, or developing fruits. Because the location of sources and sinks changes throughout a plant's life, phloem transport may be upwards or downwards. You'll also remember from GCSE that root hair cells have a large surface area to absorb water and minerals from the soil. Water enters the root hair cells by osmosis, while mineral ions are often taken up by active transport, particularly when their concentration is lower in the soil than inside the root cells. At A level, you'll discover that getting water from the roots to the leaves is far more remarkable than it first appears. Water can travel tens of meters up a tree without the plant having a pump. Rather than being pushed upwards, water is effectively pulled through the xylem by the evaporation of water from the leaves during transpiration. You'll learn how the cohesion between water molecules and their adhesion to the xylem walls make this possible in what is known as the cohesion tension theory. Now, you'll remember that the rate of transpiration is affected by several environmental factors, and it's well worth revisiting these before you begin Year 12 because they're some of the first ideas you'll build on at A level. Let's start with light intensity. As light intensity increases, the stomata usually open wider to allow more carbon dioxide to enter the leaf for photosynthesis. With the stomata open, more water vapor can diffuse out, so the rate of transpiration increases. You'll also remember that temperature has an important effect. As temperature rises, water molecules gain more kinetic energy, so they evaporate from the mesophyll cells more quickly and diffuse away from the leaf faster. And this increases the rate of transpiration also. Humidity has the opposite effect. When the air is very humid, it already contains a lot of water vapor. So the concentration gradient between the air spaces inside the leaf and the surrounding air is reduced. As a result, water vapor diffuses away more slowly and the rate of transpiration decreases. Now, finally, wind speed. Moving air removes the layer of moist air around the leaf, maintaining a steep concentration gradient between the leaf and the atmosphere. And this allows water vapor to continue diffusing away rapidly, increasing the rate of transpiration. Now, if those explanations sound familiar, brilliant. They are exactly the foundations you'll need at A level. The difference is that you'll be expected to explain these ideas in greater detail and apply them to unfamiliar situations. You may also remember carrying out or at least learning about the Potometer. This practical becomes much more important at A level because it's one of the pieces of equipment named on the apparatus and techniques list on all exam boards. A potometer is used to measure the rate of transpiration by measuring the rate of water uptake by a cut chute. Over a short period of time, we assume that almost all of the water taken up is eventually lost through transpiration. So, water uptake provides a good estimate of transpiration rate. Now, one common misconception worth correcting now is that a potometer does not directly measure transpiration. Water uptake is an estimate of transpiration. Some water taken in can be used in photosynthesis or lost through the seal between the stem and potometer. Now, as you move into year 12, you'll also think much more carefully about experimental design, identifying variables, justifying methods, reflecting on exactly how we can design studies to give accurate, reliable, and precise data, suggest improvements to reliability, and also evaluate the limitations of using water uptake as an estimate of transpiration. Finally, you're also going to build on your understanding of leaf structure at A level. You'll probably remember from GCSE that a leaf is specially adapted for photosynthesis and gas exchange. Rather than simply memorizing the different tissues, try to think about how each one contributes to those functions. So, starting at the top of the leaf is the waxy cuticle. This transparent waterproof chemical layer, not a tissue, helps reduce water loss by evaporation, while still allowing light to pass through to the photosynthetic cells below. Beneath this is the upper epidermis. These cells are also transparent, allowing as much light as possible to reach the palisade mesophyll. The palisade mesophyll is packed with chloroplasts and is the main site of photosynthesis because it lies close to the upper surface of the leaf. These cells receive the greatest light intensity, have a high concentration of chloroplasts, and they also tightly pack together. Below the palisade layer is the spongy mesophyll. These cells are more loosely arranged, creating large air spaces between them. These air spaces allow carbon dioxide to diffuse rapidly to photosynthesizing cells and oxygen to diffuse away. Running through the leaf are the vascular bundles containing both xylem and phloem. The xylem transporting the water and mineral ions into the leaf and supplying the water needed for photosynthesis and replacing water loss through transpiration. The phloem is transporting the sucrose and other molecules away from the leaf to areas of the plant that require them. And then finally, on the lower surface of the leaf is the lower epidermis, which contains most of the stomata. Each soma is surrounded by a pair of guard cells that control whether the pore is open or closed. Rather than simply recognizing stomata as pores that allow gas exchange, you'll explore how guard cells regulate stomatal opening, balancing the plant's need to absorb carbon dioxide for photosynthesis with the need to reduce water loss. At GCSE, it's often enough to identify these tissues and describe their functions. However, at A level, you'll be expected to explain how these adaptations increase the rate of photosynthesis or transpiration and to link them to underlying biological principles such as diffusion, concentration gradients, and water potential. Now, one misconception that's worth correcting before you begin year 12 is that photosynthesis doesn't only occur in the palisade mesophyll. While the palisade cells are the main site because they contain the highest density of chloroplasts, the spongy mesophyll cells also contain chloroplasts and therefore also carry out photosynthesis. So, before you move on to year 12, make sure you are confident on all of the content covered here. If those foundations are secure, you're ready to dive deeper on your A level journey and you'll be able to apply concepts in exam questions so much more easily. So that brings us to the end of this third bridging series video. If you found this video useful, don't forget to download the free summary sheets and misconception guides from the description below. They'll help you check your understanding and avoid some of the most common mistakes students make when starting A-level biology. In the next video, we'll continue building on those foundations with another set of essential GCSE topics. So if you haven't already, consider subscribing so you don't miss the next instalment. Thanks for watching, and I'll see you in the next lesson.